Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this little notes cast, we're going to be talking about the structure, and basic function of the good old cell membrane. So important that it has another name. You'll see on the Regents exams and in some textbooks and other places, people refer to it also as the plasma membrane. You'll see a lot of artists' diagrams, basically, of the cell membrane. I wanted to show you some electron microscope pictures here so that you can clearly see the two layers here of the cell membrane, that it is a lipid bilayer like we have identified it in the past. I like this diagram over here because it clearly shows how there's a different environment on one side of this lipid bilayer as there is on the other side of the lipid bilayer. All right, so two layers of lipids makes up the cell membrane. Here's one of those artist pictures that I was telling you about, and here's one layer of lipids. Here's the other layer of lipids. All right, the little hairy parts, those are the fatty acid chains that we'll talk about here in a second. So we've got two layers of lipids, but also floating around in there are all these proteins, right? Some of the proteins have big carbohydrate chains attached to them. We'll talk about those in a minute too. All right, but really what we have is we have a whole bunch of lipids, a field of two layers of lipids with some proteins and carbohydrates floating around in it. You can't see them in this diagram. We'll show it in another diagram, but in amongst the legs here of the phospholipids, there are some cholesterol molecules. All right. The job of those cholesterol molecules is to keep these legs from getting too kind of packed together. It keeps them kind of disarrayed a little bit. To look a little closer at the phospholipids that are in the lipid bilayer. If you remember, a basic triacylglyceride is made up of a glycerol with three fatty acids that are attached, and here we're looking at a phospholipid. So the phospholipid has two fatty acid chains attached to the glycerol, and instead of a third fatty acid chain, it has this phosphate head. Now, this phosphate head is going to give the top part of these phospholipids a different characteristic. It makes them what we call hydrophilic. Hydro for water and philic for loving. So these sides here, these heads of these phospholipids really love to be in water. The fatty acid chains here are they are all carbon and hydrogen, makes them hydrocarbons. Hydrocarbons are nonpolar and therefore don't like water, and we call that hydrophobic, for fearing water. So the tails are constantly trying to get away from the water, where the heads are trying to get into the water. All right, here's an individual phospholipid right here. So what's actually holding these lipids in place is the fact that the heads like to be in the water and the tails like to be away from the water. They like to be with other tails. All right, and this arrangement kind of spontaneously sets up. All right, it's kind of cool. So those individual phospholipids aren't bonded to one another, so there isn't a rigid or a hard connection between these two. All right, all that's really keeping them together are the hydrophilic, hydrophobic ends trying to get in and out of the water. This lack of bonding between one another allows the membrane to kind of flow. It's a dynamic structure, right? So these can kind of form like waves, like ripples almost through here, because these guys can kind of jostle around a little bit. This is actually the same way that soap bubbles work. A soap bubble, or a soap molecule, is made out of a single fatty acid chain that has a sodium group or a phosphate group, a big, huge polar group up here at the top. And it works the same way. The phosphate group of the soap likes to get into the water. The hydrocarbon tail of the fatty acid likes to go into the fat. And so you can get a glob of fat that's just surrounded by soap molecules, so all the little heads are sticking out, and that makes that glob of fat able to be dissolved in water. All right, so it's kind of the same arrangement. There are a lot of connections. There's a lot of analogies between soap bubbles and cell membranes. The fact that you take a soap bubble and make it form two, right, is kind of like mitosis. There's a lot of things you can do with soap bubbles that act a lot like cell membranes. Here's another shot here showing you those carbohydrate chains on the outside, or carbohydrate trees. Some of my students refer to them on the outside. Sometimes people refer to these as receptor molecules. They're not really receptor molecules because they don't really receive things per se. What they are is they are really name tags. All right, they have particular shapes, and my receptor molecules are going to be slightly different from your receptor molecules, and that's how part of how our bodies can recognize our own cells versus another organism's cells. The proteins are going to form these transport tubes. Some of them, we'll talk a lot about the transport tubes. Others of them will form the receptor molecules themselves, the binding sites, the places where some of these carbohydrate chains from other cells can kind of fit in. All right, and that's how cells can. That's how cells talk to one another. They use Braille because they're blind. So these guys have particular shapes, and another cell will come up and see if the shapes fit into the receptor sites, and if they do, then the cell knows that it's self, and if they don't, then they know that it's non-self. And so this is how they can tell self from non-self. Those receptor molecules have different shapes, and the signaling molecules, the glycoproteins, have different shapes as well. All right, this is just another shot showing you all those different shapes. All right, so what do all the shapes of different receptor molecules allow the cell to do? Well, like I said, they act as identifiers so that you can, the cell can tell self from non-self. All right, this is how your immune system is going to work. It's also going to allow the cell to recognize if what it bumps into is a useful material, is food or not, something that needs to be ingested or taken into the cell. Does it have the right shape? Okay, we'll eat you. If it doesn't have the right shape, well, maybe we won't eat you, or vice versa. All right, and like I said, this is how the cells are able to communicate with one another through these receptor molecules. One cell can send out a signaling molecule, like a neurotransmitter or a hormone or something like that, and if it fits into the receptor molecule of a neighboring cell, then that cell is stimulated to act in a particular way, to change or move or something like this. Yet another shot. This one is showing the cholesterol molecules here in amongst the legs. So we don't want these legs to nest together too closely and get too packed together, because if it does, then that reduces the flow here of the cell membrane. So we want to have something in there that's going to keep these legs from nesting together. One of the fatty acid chains is unsaturated. And the unsaturated fats, you remember, because they have the straight chains, they can pack together a little more densely and form solids. And we certainly don't want your cell membrane to become solid. The other fatty acid chain is unsaturated. It does have a little kink in it, and so that allows it to not nest together very well. So we got to kind of walk that fine line of having enough nesting together so that the cell membrane still stays intact, and doesn't force itself apart, but not so much nesting that it becomes a solid. So the cholesterol in here kind of helps keep this from becoming a solid. It makes it nice and flowy. Here's another shot that shows, I think, a pretty good view of the binding sites here. Here's the receptor molecule. Here's one of those signaling carbohydrate trees on the outside. And here's a transport protein allowing material to cross through the membrane. It's got some other stuff in here. We got some proteins on the inside, right, attached to these great big long protein filaments, all right, all important parts of the cell being able to respond to changes in its environment. So one of the main functions of the cell membrane is transport, moving material in and out of the cell. But remember, it's got to maintain a difference inside from the outside, and so it can't let everything just flow freely in and out of the cell. It's going to block some of the stuff from being able to flow freely. It's this semi-permeability, the fact that some material can pass through and some material can't pass through, that maintains a difference inside of the cell from outside of the cell. So we say that the cell membrane or plasma membrane is semi-permeable. To permeate means to just spread out. So when you open the bottle of perfume, right, the smell will eventually permeate the entire room. And really what we're talking about the smell permeating, is we're talking about diffusion. So we're going to spend quite a bit of time later on talking about diffusion and how things spread out like that. Other material wouldn't be able to diffuse through the cell for a variety of reasons, but it still needs to get in or out of the cell. And so that material is going to be actively transported or pumped through the cell membrane. We're going to use those protein channels to do this. Other materials still can't get through. It's just blocked. And there is other material that shouldn't get through, but it has the right shape, and so the cell membrane lets it in. All right, remember, it's kind of like Braille. These cells can't see each other. So if something presents the proper shape to be allowed into the cell, then it's going to be allowed into the cell, whether it's food or a virus. So to just summarize, the main function of the cell membrane then is to maintain the amount of chemicals, the inside environment of the cell, to keep that stable. All right, and this life process could also be described as regulation, because we're maintaining that homeostatic balance, chemical balance on the inside of the cell. It's going to do this by affecting which materials can go in or out of the cell, right, affecting the permeability. How does it do this? Well, we're going to spend some more time on this in future classes. Just to preview what we're going to talk about here next class and in other classes, how the material actually is forced into the cell. One of the ways, since we are talking about receptor molecules and the structure of the cell membrane, this is an appropriate time to at least introduce the idea of receptor-mediated endocytosis. Endocytosis is a mouthful, all right, but the term endo, the prefix endo here, means inward. Cyto, we've already talked about, that means cell. And we can just think of cis as like a process or something like that. But basically what this means is it's an inward cell process that is mediated or helped along by those receptor molecules. All right, so the receptor molecules are going to kind of determine whether this inward cell process is going to begin. All right, so what happens is the cell bumps into a larger molecule or a chunky bit. Right, and if that larger molecule fits into the receptor molecule on the outside of the cell, it's going to cause the cell to change. And what the cell does is it actually starts to move and surround the large molecule or chunky bit. All right, and the cell membrane actually pinches off, forming a little vacuole on the inside, and the large molecule or chunky bit is now on the inside of the cell in a vacuole. All right, this process, when it's a huge chunky bit like a bacteria or something like that, is called phagocytosis. All right, phagy is the prefix for eating or consuming. All right, cyto for cell, and cis. So this is cellular eating when it surrounds or engulfs a big chunk. Pinocytosis is referred to as cell drinking. This is when the cell surrounds bits of the external environment, the solution that it's floating in, and it gets the dissolved materials as well. I will show you an animation in class when we get to this, but I'm going to stop right there for now. It's going to make it a lot easier when I show you this in animation when I can show it to you in person. Thanks for checking in. Again, this is Mr. Ulrich from MrUlrichsLandofBiology.com, and we'll see you in class.